Okay, I'd like to introduce you to Alan Lewis from Medistem.
Yeah, thank you, and thanks for staying later on to hear the presentations. Well, we're not talking about soybeans, and we are fiscally responsible too. But let's see, I'll use this, I guess. We are quasi-public companies with a safe harbor statement. The focus of the company, we are a clinical stage virtual company. There's only a few employees, and we have a singular focus right now on a population of stem cells called endometrial regenerative cells, which I'm going to share the rest of the presentation about. These, I'll call them ERCs for simplicity, and they are derived from the endometrium, which is the lining of the uterus, I'm sure you're all aware, and that we call them nature's regenerative powerhouse, because women go through about 500 cycles in their lifetime here, and these cells can keep regenerating. And the cells in particular, the ones that I'm talking about, the ERCs, are very specialized in making blood vessels. They're very angiogenic. They're also pluripotent, so they can actually have broad applicability. You can see they actually can differentiate in a variety of different cells in the body. But it's their angiogenic activity that I'm going to focus on this afternoon. Because they're from the category of stem cells which are called mesenchymal stem cells, they have a universal application. They're allogeneic. And they are safe, and I'll again share more about that. We do have FDA approval for manufacturing as well as clinical trials, and it's a very efficient way of generating cells using this particular cell population. One donor can generate as many as 20,000 different doses of cells, and because they're pretty sturdy cells, they're frozen, they can be sent across the country, around the world, as a point-of-care product, like a pharmaceutical model, if you will. The initial indications that we're going after are very large, both cardiovascular indications, CLI, critical ischemia, as well as congestive heart failure. And we also have additional possibilities, which I'll share with you. Very important for little companies is IP, as you all know, and we have a very extensive intellectual portfolio, which again I'll mention a little bit later. So these cells were discovered by Tom Ickham in 2007, shown to be very novel cells, pluripotent, and of course they were obtained from the endometrium. And you can see from the cartoon that they are truly pluripotent. They move into most cell types in the body. These cells were independently verified by a number of other labs around the world, actually. So I think we know that these cells are in existence. You also notice that the publication date for these publications are 2008. So we do have the dominant IP in this space. So this slide shows you that the endometrium on the uterus regenerates itself every month. And it's really a very, very vascular tissue in the body. And it's the ERCs that are these specialized cells that are critical to produce blood vessels within that tissue. And so the term regenerative powerhouse, it is one of the very few organs in the body which has that kind of recycling. So the question, of course, is, well why do they cause angiogenesis? And this simple slide just conveys how we believe it does this. When you inject these ERCs into ischemic tissue, whether that be limbs, heart, or other organs, it turns on the protein HIF1 alpha, which is an important hypoxia-induced factor. This protein then actually activates growth factors, angiogenic factors in particular, as well as matrix metalloproteinases in the ERC. And between those two proteins, you actually will then generate blood vessel formation. So this is a unique way of generating blood vessels. Now we know that the ERCs are, I use the word unique carefully here, but this work was done at the NIH, and they wanted to compare gene expression profiles of bone marrow mesenchymal cells, which is perhaps the gold standard in this space, with our ERCs. And you will notice on this slide that we have a multifold upregulation of genes on the slide, from 39-fold for aldehyde dehydrogenase to, going right down to smaller fold increases. But the thing I wanted to point out is that in light blue on the slide are those proteins, those genes, which are linked to angiogenesis and clearly blood vessel formation. Some of the other genes on the slide are linked to what I'd call the anti-inflammatory profile of MSCs, and clearly there are a number of MSCs that are in development for the purpose of suppressing autoimmune diseases. So the genes are regulated, so what happens in vitro in a cell dish? Well, this is a slide that just shows you that these ERCs will simulate blood vessel cell growth in vitro. Here we're measuring endothelial proliferation and different concentrations of ERC supernatant. And then in the bottom of the slide, we've also shown quite convincingly that ERCs actually simulate angiogenesis these are in mice. What you do is you actually clamp off the femoral artery, and on the left hand, as you look at it, you see what happens over a number of weeks. You lose the limb. If you do the same procedure with the mice on the right and give them these ERCs, you can see the limb is retained. So quite a obviously a little gory slide, but it shows you that these cells do have very significant in vivo activities. Also, this is a model of heart failure, and in a similar process here, we clamp off blood vessels, LAD, and you'll notice after 14 days of the clamping, you then administer the cells, or the cells could be bone marrow in this case, or ERCs as well as controls, and measure what happens 14 days later. Now you get significant fibrosis, which is the dark coloration on the slide, and you will notice in the histogram that the ERC is much more effective at reducing fibrosis of the heart. And if on the second figure on the slide, you'll also note that the left ventricular fractional shortening is increased after the administration of ERCs, but not the bone marrow cells. Again, showing that cells do function and they reduce scar tissue as well as increase tissue repair. Obviously, moving right along the process, we wanted to make sure that these cells were safe in order to go into the clinic, and we've shown that they indeed do not demonstrate either acute or chronic toxicity in animals at doses exceeding 100-fold what you'd expect to dose humans, and this is over a period up to 12 months. The FDA has accepted this clinical package, and they're allowing us to move ahead with our clinical trials. The other thing, of course, I'm sure many of you realize that angiogenesis has been linked to tumor formation, and clearly we needed to check that out, whether in fact these cells would enhance tumor formation. So we used an interesting model of glioma in mice, and the pink on the figure shows the brains of mice, and in blue you can see the actual tumor itself. And at the top you see the controls, and then if you go down you'll see that the ERC is administered intravenously, as well as given directly into the tumor site. You'll note that the actual cells reduce the tumor formation in these animals. Now, we've obviously looked at a number of patients to date, and not looking for tumors directly, but I think it's a good sign that we're not going to see anything. And I think the other thing is, I wanted to point out that even though they're angiogenic, these cells really are angiogenic in ischemic situations. So what is the diseases that we're going after? CLI, I'm sure people have been talking about this all day. This is one of the favorite diseases for those people working with MSCs. You know, clearly there's a occlusion of the circulation in the extremities, result of atherosclerosis, often linked to diabetes, with very significant outcomes, and you can read the numbers for yourself, but clearly there's a huge number of leg amputations, and the only treatment right now is not terribly, well, it's effective, but involves surgery, of course. And you can imagine that with reimbursement at the level that we're talking about, which is an estimate to date, this is a very significant market opportunity for the company. And again, there are no effective therapies being developed. Oh, well, I guess other companies would argue that they have good compounds moving forward, or cells moving forward, but we believe ours are specifically interested because of their angiogenic potential. Currently, we've got an FDA-approved phase 1/2 trial, which will be initiated in the first quarter of next year, with 15 patients which are really are not eligible for surgical revascularization. The PI is Mike Murphy, who some of you might know at Indiana University. He's the very first person to have done a bone marrow stem cell trial in CLI in the United States. We also have a very small trial ongoing in China for a CLI study with a small group there. it's commenced earlier this year, and we've only done 2 patients to date. No issues, but obviously there's no data either at the moment. So this is a sort of a global approach. It will give us some information which could be very useful. Moving on to the CHF studies, again, a very large opportunity, marketing opportunity. I won't walk you through the numbers here. Significant mortality rate. The fact of it is, again, there isn't really much going on which is going to really change this. And we've already initiated a phase 1/2 trial earlier this year, which we will complete in 2014. It's 60 patients. These patients are eligible for CABG and angioplasty, as you can see. We use a procedure whereby the cells, the ERCs, are administered in a procedure which involves retrograde infusion of cells into the venous sinus, shown in blue on the slide, with the help of a balloon catheter, which creates a black pressure, so the cells are allowed to go into the tissue at that point. It's a 30-minute procedure, and there are a number of companies that have much more convoluted and more difficult procedures in place at the moment. We've done 13 patients to date. Again, it's a double-blind trial, so I can't share data with you. We do know that the DSMB has approved continuation of the study after the first 5 patients, so we haven't seen any real issues from a safety perspective at this point. Again, we're working with Leo Bakaria is the chairman of the Bakulov Center, that's in Moscow, a big cardiovascular center there. Amit Patel is our international PI, obviously based in Utah. So, just to give you a little snapshot of the products that we have. It's the same product, of course, but the potential of the ERCs. You can see that the congestive heart failure and critical ischemia moving forward. I have not mentioned other indications today, but again, in that one slide I shared with you, these cells are like other MSCs. They can be used in autoimmune diseases. We've got some animal data in this area, but we're not going to be talking about that. In MS, Duchenne's, which of course is an orphan disease, and an area which I'm particularly interested in is type 1 diabetes. This is an autoimmune disease I'm sure most of you are aware that has been a challenge to cure, but my old company ViaCyte is doing well with an embryonic program. A few people have tried MSCs to see if they can suppress the immune system of these patients. So not terribly effectively to date, but I think our cells are slightly different, and we're working with Yale University, Hugh Taylor and his group, Bob Sherman, to come up with a program which we think will be a very important one in this space. So overall, the opportunity for all these types of indications is enormous. I'm not telling you what you probably have heard of multiple times today, but our current approach is on the 2 programs which I mentioned at the beginning. The IP, Tom Eicham is a true entrepreneur, innovator, and an outstanding person